At the Human Islet Isolation Laboratory, a donor pancreas has been received for pancreatic islet isolation. Under sterile conditions, the pancreas is prepared. Blood vessels and excess fat tissue are removed. The main pancreatic duct is identified. The duodenum and spleen are also removed. Approximately 1 million pancreatic islets are dispersed throughout the pancreas. The islets, depicted in red, are surrounded by exocrine tissue, depicted in green. Insulin-producing beta cells are the main hormonal cell type in the pancreatic islets. To continue islet isolation, a small catheter is inserted in the main pancreatic duct. Subsequently, an enzyme solution containing collagenase is infused through the catheter. Collagenase that leaks out of the pancreas is reinfused. Meanwhile, a digestion system is prepared. This system contains a digestion chamber with iron marbles. The pancreas is cut into pieces, which are put in the chamber, and a lid is attached. A warm solution is pumped through the digestion system, including the chamber. The chamber is shaken so that the pancreatic islets can be freed from the exocrine tissue. Samples are taken from the recirculation solution to monitor the digestion process under a microscope. If the islets are free from the exocrine tissue, islets are collected in conical tubes. The conical tubes containing the islets and small pieces of exocrine tissue are placed in a cold centrifuge. The digestion solution is removed and a cold washing solution is added in order to stop the digestion process and protect the islets from the collagenase enzyme as much as possible. The washing procedure will be repeated and the digested tissue is put in another solution for islet purification. In order to purify the islets, a continuous density gradient is made using a high density and low density solution in the spinning compartment of a cell separator machine. This density gradient is important to separate the lighter islets from the denser exocrine tissue. By reversing the flow in the cell separator, fractions can be collected that are either enriched for the islets or contain mainly small pieces of exocrine tissue. The fractions containing islets with acceptable islet purity are put in a transfusion bag. In this way, there is a considerable tissue volume reduction while using the majority of the islets for transplantation. The transfusion bag containing the islets is placed in an incubator. Safety testing for bacterial contamination and function testing for glucose-stimulated insulin secretion are performed for quality purposes. If the islets are released for transplantation, the transfusion bag with the islets is transferred to the radiology department where the islet transplantation takes place. Under local anesthesia, a radiologist performs a percutaneous transhepatic catheterization of the portal vein under ultrasound guidance. A catheter is inserted into the portal vein. The transfusion bag is connected to the catheter. The pancreatic islets are infused in approximately 20 minutes. The islets embolize the small branches of the portal vein without major adverse consequences. In fact, the liver has now adopted pancreatic islets and the islets start producing insulin in the liver of the recipient.